Okay, anatomy students, this is our review on cellular organelles, cellular activities, functions, interactions. All right, so this is just a kind of boring summary of lots of organelles. You probably have most of it in your notes, do that in your own time. This is simply a little cell labeled cell. One thing to note is that there's no vacuoles in animal cells, so we're not studying vacuoles. Instead, we talk a lot about vesicles, transport vesicles, and we'll talk a whole lot about them. They look like lysosomes insofar as they're just spherical, double membraned little packages. So they're easy to mix up. Notice also the difference between the rough ER and the smooth ER, very much the same. But the rough ER is studied with ribosomes. Okay, this is the usual cutesy biology, as I call it. Come back to this at the end and make sure you can identify all of these. I think many of them are pretty easy. Okay, let's start with one organelle at a time. The nucleus contains all the genetic material, as you know, and second bullet point, it directs all cellular activities. I have a slide for each A and B here. So what does the nucleus do? This is cell division, which we're not studying in depth, but you remember that for cell division to occur, what has to happen? Look at each of the daughter cells has a full copy of the DNA. So the genetic material has to be copied, and that occurs in the nucleus. Secondly, if you do not remember this, you probably need to go back to it and review it very quickly. Inside the nucleus, in this image, this is the nucleus here. Every time something is needed, every time a protein is needed, which is very often, a huge number of proteins are being made continuously in your body, the DNA, the correct section of the DNA is found, this is a section of the DNA that has the instructions for the needed protein. An mRNA copy is made. Here's the mRNA copy, and it's sent out into the cell. I'll remember what this organelle is in a moment. Sent out to the ribosome, so that that mRNA copy, which is essentially the instructions for a protein, will indeed become a protein. Okay. This organelle is easy to recognize with this distinct shape and distinct texture in the middle. What's important about it is in cutesy biology, it's always called the powerhouse of the cell, and everybody seems to remember that. You guys have learned more, and now you can say it's the place where which chemical reaction occurs? Cellular respiration. Cellular respiration is a chemical reaction in which nutrients are broken down and energy is released. Okay, so it's like the second part of digestion. Digestion occurs in your stomach and intestines. The second part occurs in every single cell at the mitochondria. Okay. I don't know why. When I'm doing a screencast, I often jam. Okay, here we go. So the lysosome is simply a little spherical package full of, oh dear, full of digestive enzymes. Digestive enzymes literally because waste materials in the body, bacteria, viruses, all kinds of bad stuff, are digested. So lysosomes, a silly way of putting it is I always say it's death by digestion, right? So in cutesy biology, the association is Lysol, which kills off germs and bacteria. So it's kind of a silly association, but for those of you who are helped by cutesy biology, you should recall this. We talked about this. This is a white blood cell, WBC, a white blood cell, which different kinds of white blood cells kill in different ways, but this one is one which its method of attack is death by digestion. Here's a virus that it engulfs through endocytosis. And what are these? They're little lysosomes. What are these black thingies? They're digestive enzymes. And so it's going to kill the virus and spit out its contents. So death by digestion, this white blood cell has many lysosomes in it. Okay, so now we get to some more important organelles, or more interestingly complex organelles. This is the ribosome, right? You recall it looks like just a tiny dot in the diagram of the cell. It's just one tiny little black dot usually. And here we're zooming in on it. And what do we see is happening? This all-important green ribbon is the mRNA. It's the instructions for making a protein. This is the beginning of a protein. It's a polypeptide chain that's being churned out as the ribosome and the tRNA follow these instructions, okay? If you're confused by this, you can review this worksheet on the unit, we covered this, or a really good summary is in the video from DNA to protein. And remember, I pointed out that order is all important here, the order of the nucleotides signifies what order the amino acids will be, and that's what makes the protein what it is. Okay, now we get to endoplasmic reticulum. So endoplasmic reticulum is this crazy, strange networking stuff. Notice these are, the interior part is light blue. The darker gray blue signifies the exterior. Notice there's also two types, the smooth one and the rough one. The rough one isn't truly rough, it just is coated with lots and lots of ribosomes, right? Let's define the two different types. Okay, so if you want a quick definition before we go into it, rough ER is involved in protein folding, protein formation, protein modification. Remember, there's lots of little modifications that are made, protein sorting, protein transport. So it's all involved in making proteins after the ribosome has spit out the first level of protein, the polypeptide chain. The smooth ER does the same thing, but for lipids. And so if you think of it, it is the place where our phospholipids are made, where our cholesterol is made. So since both of those are found in the cell membrane, you might notice it's involved in membrane synthesis. It's also involved in the detoxification of harmful chemicals. Okay, let's look at this picture first. This picture is the ribosome, in which place the first level of protein is being made. But look at this. Let's get a little closer look. Most ribosomes, here the ribosomes are yellow, not brown, are found on the surface of, can you guess what this is? This is rough ER. And in fact, through a little pore of the rough ER, the ribosome is sort of churning out, spitting out its protein into the empty part of the rough ER. So the rough ER is receiving the protein chain. And if you notice, the folding, which is such a big deal in proteins, if you remember, the folding occurs inside the rough ER. Okay, so here's another picture of the same thing. This is the same thing diminished here in this picture. Same thing's going on. We've got one ribosome. It's brown now again. And they didn't show us the mRNA, but we know it's following the mRNA instructions to make a protein chain. Here's the protein chain. And inside this big blue thing is a piece of rough ER. Inside the body of the rough ER, the protein chain is being folded. It's being modified. These little green carb chains are being added. And eventually it's going to be packaged and sent off. Okay, so now this is the bigger picture. And let's look. So here was our protein being made. Then it was packaged. And where is it sent? This shows us the next step. So we left the rough ER and we're arriving at the Golgi apparatus. That's our next organelle. So here we are, the Golgi apparatus. And its definition will sound a lot like that of rough ER. It's just the second part of making proteins, modifying, sorting, and packaging of proteins, as well as of lipids. So it receives the lipids made from the smooth ER, the proteins made from the rough ER. It receives them in little transport vesicles. So here is a protein made in the rough ER. It gets packaged and brought over to the Golgi apparatus. Here's an incoming one. It's going to fuse here. It's going to be modified, sorted, whatever is going to happen to it. And at some point, it's going to be completed, and it'll bud off and go off to its final destination. So there's lots of little transport vesicles fusing onto the Golgi bodies and coming off of the Golgi bodies. Golgi apparatus, either name is fine, really. Okay, notice in this picture, this protein that arrived here and then got modified, when it was finally secreted again, it was secreted out of the cell entirely. So it was a protein for secretion, something that needed to be given to the surrounding tissues. An example would be a digestive enzyme. This could be a cell, which is the lining of an intestinal or stomach lining, and it's making and secreting enzymes, digestive enzymes. Okay. This is the same picture. I really just want you to make sure you understand this whole process. And one more thing I want to point out, if this is being secreted by the all famous process we learned, exocytosis, you should recognize it. If not, do a quick review of exocytosis. Then that means we know the cell membrane is made of phospholipid bilayer. So the transport vesicles must be made of phospholipid bilayer. So what must the Golgi body membranes and the rough ER and smooth ER membranes be? Phospholipid bilayer. Okay, so all our work on the cell membrane is really important in lots of cellular functioning. Okay, these are good examples of what might happen. So here we are again, we've got our rough ER, our little ribosomes pumping out proteins that are being folded. What can be the result? These are free ribosomes of these proteins. So some of them will get processed by the Golgi, then they'll be secreted. Some of them, proteins secreted from the cell, proteins which might make up the cell membrane. Some of them will be proteins that are enzymes to be lysosomes, to actually become the organelle lysosomes. Some of them will be proteins that remain in the cytoplasm. Cytosol is just another name for cytoplasm, or that form the cytoskeleton. Some of them will be specific proteins that get taken to a particular organelle. This is a very useful summary of, it's called the endomembrane system. I'll talk about it in 2 slides, I think. Okay. So this is just a reminder of what exocytosis was. This is a visual image of what exocytosis was. This is your membrane-bound organelle, and here it is secreting its contents by fusing with the cell membrane. Okay, so this is step 1, step 2, step 3. These are all the same thing at different moments of time. This is exocytosis when it's being secreted, but something very similar to endocytosis and exocytosis is occurring continuously on the ER and the Golgi bodies. I call it budding off or fusing into, right? There's this continuous budding off or fusing onto the Golgi bodies and the ER. You can see it again here, right? This one's about to fuse, etc. This one is budding off. This one is just, these are just starting to bud off, and so on. Okay, I think you get it. Just want to make sure you've got a real visual image of it. Okay, here's our next question. Which organelles are part of the endomembrane system? So let's define this. Endomembrane system is the group of membranes and organelles that work together, and in that way they're a unified functional system. They work together to do what? To make, to modify, to package, to transport proteins in the rough ER, lipids in the smooth ER. They continuously exchange material through vesicle transport, as we've been talking about. So here's a good visual image of the endomembrane system. So which organelles are involved? I think it's pretty clear and pretty easy to see. The rough ER, obviously, the transport vesicles. Look at all these various transport vesicles. The rough ER, the smooth ER, the Golgi apparatus, the transport vesicles. And we could sort of add, although it's not exactly an organelle, we could say the cell membrane. Here is the exocytosis, the example we looked at before, secretion. So that was a protein that was made here, right, made in the rough ER, taken to the Golgi body for modification and packaging. It's completed, buds off, is secreted. But the same thing can occur in the smooth ER with a lipid, right? Transported to the Golgi body, modified in the Golgi body. When it's ready, it might also be secreted. It might be brought to the cell membrane to be part of the cell membrane. Here's another one which is being, which is a lysosome, and so it's fusing with something the cell has taken in by endocytosis. Anyway, you see that there's many examples of this big complicated network of the endomembrane system. You can see this video is a good summary of it. It's pretty short and it's pretty easy. Okay, so read that definition, make sure you understand this picture. That was there randomly as a review at the beginning of one class. Okay, here we go. Cytoskeleton is a network of protein filaments. And there's 2 types of protein filaments we're going to learn about, which provide the cell structure, shape, and routes for transport vesicles. Okay, so the summary is protein filaments provide cell structure, cell shape, routes for transport vesicles. By the way, these are real photographs. They put pigments which bond with certain proteins in certain ways to highlight these protein filaments so you can see them. Okay, there's 3 types of protein filaments. We're going to ignore this altogether, overlook. We're going to focus on the microtubule. Let me just flash ahead for a moment so you can get a better picture of it. This is a better picture of a microtubule. It's actually hollow and cylindrical. The microtubule is important in cell structure and shape. It's rigid. Just a quick comment, without these proteins, the cells would just be big blobs. They are routes for transport vesicles. They're involved in carrying vesicles out of the cell for exocytosis or into the cell for endocytosis. They're part of the spindle of mitosis you learned about in biology. They're part of cilia and flagella you learned about in biology, etc. Microfilaments we're going to learn less about, but they are also important in cell shape, cell division, in motion, contractile proteins. Okay, so here we go. Here are the microtubules, this cylindrical structure of proteins, and here are some of the routes and pathways, if you will, of different things being made, secreted, given to different organelles, etc., etc. These are some of the cellular activities. Each one of these is a pathway, and what makes that pathway? Microtubules.